We're delighted to bring you the seminar presented by Labroots and in collaboration with the National Institute of Health. Welcome to our track on the NIH Brain Initiative, a multidisciplinary approach to neuroscience. We're glad you can join us for this panel presentation titled Sensorimotor Processing, Decision-Making, and Internal States towards a realistic multiscale circuit model of the larval zebrafish brain. It's my pleasure to introduce to you a panel of experts who will be speaking on the topic of understanding circuits and behavior. For a full listing, please click on the tab at the top of your screen. If any questions should arise during the presentation, we encourage you to submit them into the Q&A box. Our speakers will respond back to you via email. And as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the top right corner of the presentation window and follow the process to obtain your credits. Now, please join me in welcoming our panel of experts.
Hello, my name's Florian Engert, and I'm a member of a team in the context of the NIH U19 grants that are trying to find out how the brain of a larval zebrafish gives rise to adaptive behavior. So as you can see, the title of our presentation is Sensorimotor Processing, Decision-Making, and Internal States towards a realistic multiscale circuit model of the larval zebrafish's brain. In short, what we're trying to find out how the brain of this animal, the neural circuitry, gives rise to adaptive behavior, to gain an understanding of that. And I probably should introduce the zebrafish as a model system in circuits neuroscience. And the next slide here illustrates the power, but also the challenge that this animal provides. It is tiny, it's only about 4 millimeters in length, a millimeter across, and it is perfectly translucent. So it allows us to, in principle, visualize neural activity throughout the brain of an awake and behaving animal. On the left, you see a still shot of neural activity measured across the brain. In the center is a top-down view. On the top is a side-on view. On the left is a head-on view across larval zebrafish. On the right, you see a still shot of the anatomical reconstruction of a subset of the neurons. And what will convince you over the course of the whole presentation is that we can actually combine these two technologies in the same animal. We will now show a video where you'll first see the activity patterns across the brain as you see on the left, and it will follow with a visualization of the 3D structure of some of these neurons. I'll start the movie now and allow you to appreciate the beauty and complexity of a live brain in action. Please, let's get going with the movie. You could appreciate the complexity and the beauty in those two images. For the record, I should point out that these two movies have been taken in separate animals, but the whole purpose of our collaboration here is to allow us in the future to combine brain-wide imaging, as shown in the first part, with brain-wide connectomics, i.e. the wiring patterns of the neurons that give rise to this activity in the same animal. This is will be in more detail. How we're going to do this will be in detail discussed in the down the road. In the next slide, is posed the central question. What are we going to do with all of these data? These are terabytes and petabytes of data. And how are we going to use these data to gain insight about what the brain is actually doing and why it's doing what it's doing? So I think it's important to point out that just description of the activity is not really an explanation of how the brain works. So to briefly outline how we will connect data to brain function, I want to briefly give credit to David Marr. He's a theoretical neuroscientist who died, I think, in 1981, but one of the pioneers in thinking about brains. And he allowed us to pose the question more in more detail. What does it mean to actually understand the brain? So the way he phrased it is, any machine carrying out an information processing task must be understood at the following three levels. And the three levels of understanding the brain, as posited by David Marr, are the following. The first question you have to answer is, why is the brain doing what it is doing? What is the goal of the computation? Why is it appropriate? And what is the logic of the strategy by which it can be carried out? Or in other words, what good does it do for the animal, mostly in an evolutionary perspective? The second level of understanding, according to David Marr, is the what. What's the algorithm? What's the representation of the computation? Or in other words, how can this computational theory be implemented? In particular, what is the representation for the input and output? And what is the algorithm for the transformation? And only lastly, once we have an idea of those two first bullet points, I think does it really make sense to engage more deeply in the how, namely what is the hardware implementation? How can the representation and algorithm be realized physically? So this is sort of the large line of thinking along which we have organized our whole project. Here are the three levels of understanding by David Marr, and I think we are trying to get sort of a handle on all three of them. So what's really important, as you've probably gathered, is behavioral algorithms. What are exactly the behaviors that a larval zebrafish can do? So that's one of the challenges that we have addressed in the last 10 years. And this is illustrated here in this slide, where we've outlined and depicted in cartoon form 5 of adaptive important behaviors that a larval zebrafish has to execute in a natural environment. The first is the opto-motor reflex, which I will discuss in more detail in the next slides, which basically says that a larval zebrafish, when confronted with whole field moving stimuli, will swim to follow along with the stimulus. The next is Rheotaxis, meaning if the fish is in a river or in a moving stream of water, it will generally tend to swim against the current, presumably in order to stabilize its position in a moving stream. There's phototaxis. Fish will swim towards the light when given a choice between light and dark. That's probably useful because they're highly visual animals and they like to be in an area where they can see, where they can use vision. The final two behaviors, hunting and escape, are obvious in terms of Marr level 1. Why is this useful for the animal? These are also behaviors that we're planning to analyze in more detail. I will, for the purpose of this presentation, focus on the first two, the optomotor response and Rheotaxis. I will discuss in more detail the optomotor response, the first, and Chaim Polinsky and Nimrod Shaham will then delve a little bit more deeply into the problem of Rheotaxis. Let me first discuss the OMR, the optomotor response, in a little bit of detail here. So as I've said already, it's a fish will swim along with a whole field moving stimulus. And this is shown in this little GIF. So there's a fish swimming on a normal patterned background. When the background starts to move, the fish will turn and swim along with it. This is a real fish swimming along. And the way you can think about this is that not the background is moving to the left, but maybe the fish is being swept to the right. And therefore, again, this is just an attempt of the animal to stabilize its position in a moving stream. This can be discussed at two levels. First, Marr level 1, why is this useful for an animal? And maybe it's useful because it allows them to stay in the region where the parents have laid the eggs. So maybe that is a useful and an adaptive, favorable position for a larval zebrafish. And if they're being swept along, they're just trying to counteract this, this movement. And to analyze this behavior, so to get to Marr level 2 in more detail, we are using a trick which is called closed loop behavioral analysis, where we monitor the fish with a camera, and we will update the stimulus in closed loop according to the position and the orientation of the animal. What this allows us to do is expose the fish to a leftward moving stimulus. And when the fish is moving and turning to change its direction, as shown in this animation, the stimulus is locked by the feedback loop to the fish such that there's always leftward stimulus no matter what the fish is doing. This is frustrating for the animal because it can't really escape this condition where the stimulus is always moving to the left. But it allows us to really collect now a large set of data under these very well-defined conditions. Here is a movie where this is happening in real life now. So the stimulus is always moving to the left of the fish. Fish is trying to align itself with the orientation of the motion. And again, we can run this for many, many hours and collect millions of individual swim bouts that allow us to dissect in great detail what exactly the algorithm is, what's the nature of the input, what's the nature of the output, and what is the algorithm that connects the two. Kumaresh will later give a brief description of a project where we will stimulate the two eyes of the fish with different motion and see how they integrate, how the animals integrate at the behavioral level stimuli that might be in conflict with each other. Here is another example of a behavior. In this case, it's Rheotaxis, where not the visual stimulus is moving, but the water is moving around the animal. So once the movie gets started, not quite yet, what you will see is that there's flow coming from the right. We just pump water through this tube. And what you'll see is that the fish will start swimming and orienting itself against the stream. There will be two elements to the movie. One, where the fish has visual information as well, where the light is switched on. In the second part of the movie, the second trial is where we switch off the light, and you'll see that the fish still can orient against us, which tells us that these animals can do this also in the absence of visual stimulation, which opens the opportunity to study a completely different modality, not the visual modality, but the somatosensory modality. But now I let you enjoy this movie. So if you can run the movie now, first with light and then without light. Please go. The visual input to execute this behavior, which really opens the fascinating question is what sensory modality are they using? And more interestingly, even what is the exact algorithm that they're using to execute this behavior? Nimrod later on will delve into much more details about this. So maybe I've given you a brief flavor of what the whole project is about. I've discussed the three levels of David Marr. The technological approaches we have, we can do imaging, as shown on the upper left, meaning calcium imaging of brain wide activity. We can image the whole animal and get a readout of the behavior. As I've said already, we are collaborating with a group that is also is focusing on the theory, so we can use theory to extract the behavioral algorithms. And that also will inform the circuit theory that allows us to think about how the neural networks that make up the zebrafish's brain give rise to the behavior. And finally, we can use connectomics, so EM-based anatomy, to check, validate, constrain, and explore the circuits further that are being proposed and put forward based on brain-wide imaging of the neurons and behavioral theory. Here are the two team leaders. On the top is Florian Engert, bottom right, Chaim Polinsky, and bottom left, Jeff Lichtman. And I think it is really a beautiful example of a collaborative enterprise where different expertises complement each other in a very productive and meaningful way. So Marr level 1 would be mostly the behavior, allows us to speculate about the evolutionary reasons why this behavior arises. Marr level 2 is best addressed with behavioral theory, whereas the other technologies allow us to dissect in detail the implementation at the neural circuit level. The whole team is much more than just the 3 PIs. I think we mostly stand there and applaud, and maybe occasionally direct, but it is a large-scale enterprise and that we believe is necessary to combine all of these different technologies. The 3 aims we are trying to address in the project is first to find a holistic theory that includes behavioral algorithms with network implementation for these 5 behavioral modules that I've described in some detail. Aim 2 is to ask what happens if the fish is exposed to conflicting or parallel sensory stimuli. In the real world, larval zebrafish don't have the luxury of dealing with OMR, phototaxis, hunting, and looming, predator avoidance in sequence. They usually have to deal with a multitude of sensory stimuli that bombard the fish in parallel. So aim 2 is of our project is to find out what happens if the animal is exposed to conflicting stimuli. And aim 3 is the question how these sensorimotor transformations and decision-making processes that we are describing here, and how all of these processes are influenced by internal states, meaning if the fish is hungry, if it's tired, if it's stressed, if it's in danger, if it's lonely. There are many neuroendocrine and neuromodulatory processes that put the animal into different internal states. And we want to know how these internal states, these slow dynamics, modulate the fast processes that we are describing in aim 1 and aim 2. So it's not just the outer fast sensory motor processing outer brain that we are interested in. We are also interested in the inner brain that reflects the internal state. With that, I'll finish my presentation and I will hand over to Chaim Polinsky, who will lead you through our efforts of trying to get a theoretical and algorithmic concept of all of these.
Hello, my name is Chaim Polinsky. I'm a theoretical neuroscientist participating in the Zebrafish U19 team. My role is to build computational models and mathematical theories of the behavior and the underlying neural circuits in order to uncover brain-wide principles underlying sensory motor functions. The first project is about optomotor response, the tendency of the animal to stabilize itself with respect to its visual environment. In the first phase of our collaborations, we have constructed a draft brain-wide neural circuit model of the OMR, as you can see in the top right of the slide. This, however, is a coarse-grained model describing the interaction between large groups of neurons. To develop a high-resolution neural circuit model, we need to challenge the animal and its brain with complex visual motion cues. Here we focus on scenarios where the 2 eyes receive conflicting motion cues. We ask first, what is the fish strategy to resolve such conflicts? And second, what are the neural mechanisms that implement such strategies? Ongoing efforts to address these questions will be described by Kumaresh Krishnan, a talented PhD student engaged in both experiment and computational modeling of the OMR.
Hi, I'm Kumaresh Krishnan, and I'll be talking to you about how we are focusing on integrating information from both eyes in the context of the OMR behavior in larval zebrafish. So how can we target each eye independently? We want a way to provide a different motion cue to each eye. And as you can see in this picture over here, the zebrafish can be stimulated with different directions of motion in each eye, and they will carry out the optomotor response and give us a sense of what they think is the direction of whole field motion. This gives us a sense of how they're able to integrate information from both eyes. Now the question is, how do we cover all the different directions? And in order to do that, we can sample the entire stimulus space into 8 different directions, and we present all combinations of these directions to both eyes. So through this experimental effort, what we want to address is how the left and right responses are able to combine to form the whole response. How does the zebrafish respond to motion cues in different eyes? Well, they turn with a specific angle in response to different stimuli. Over here, I show you one example where the 2 angles of motion seen in each eye are represented by the arrows on top. And the plot as such shows the distribution of turns made by the larval zebrafish. As you can see, there are a large number of forward turns, which we expect because the zebrafish wants to move. And in addition, there are these 2 side peaks on the left and right, which indicate the instances where the zebrafish turns. So we look at the distribution of turns as the response to all these stimuli, and we go ahead to try and build models of this. I will show you another case where the zebrafish is responding to a stimulus where there is only rightward motion. As you can see, in addition to the central peak where the forward swims are high, there is also an increased rightward swim frequency. And this is what you would expect when the stimulus is more rightward. In both these stimuli, you can see that the left eye sees only rightward motion, but simply changing the direction of motion seen in the right eye was able to produce such a big change in the response. So essentially what we've captured here is the likelihood of the zebrafish turning with a specific angle. How can we proceed to model something like this? Well, we make use of the observation that there are 3 components to the response: a forward component, a leftward component, and a rightward component. And we try to use this in the context of what we call the Gaussian mixture model to model this behavior. So the basis of this model is that each of these components can be characterized by a Gaussian distribution, and these 3 Gaussians, corresponding to the left, right, and the center, are combined with different weights. So the 3 different components are able to be represented as Gaussians, and this is an example of how our model represents these stimuli. This is the same example that I've shown on the previous slide where there are conflicting motion cues in each eye. And as you can see, the orange line here shows how our model is able to accurately capture the response of the zebrafish. You can see that the forward swim is captured and the 2 side peaks that correspond to the left and right turns are also captured. So in a sense, each Gaussian is modulated by a corresponding weight, and they all are referring to the turning capability of the zebrafish, either forward, leftward, or rightward. What I want you to note here is that this is just the likelihood of turning, essentially a probability distribution. This loses out any information about what the overall response to the stimulus was. In order to address this, we need to have a scale factor for each of these responses that can kind of serve as a way to denote the overall response rate of the zebrafish. I will show you one mathematical equation, which is basically what the entire modeling effort is. The first term, R of theta L, theta R, refers to the response rate. This is a measure of how the zebrafish responds to different stimuli, a different stimuli being an angle shown in the left eye, theta L, and an angle shown in the right eye, theta R. And this will be able to scale the probability distributions that we model. And how do we model the probability distributions? There are 3 Gaussians, each with their own means, mu, and their variances, sigma square. And there are 3 Gaussians here, one for the left, one for the center, and one for the right. and all of them are combined with these weights, WL, WC, and WR. Since we want them to be a probability distribution, the only other constraint that is imposed over here is that the weights sum to 1. This is, in a sense, the entire mathematics behind the modeling. Now, given that we're able to model the behavior so well, how do the weights, or let's say even the response rate R, vary across the stimuli? Let's take a look at that now. This picture shows you how the response rate, or the overall response rate of the zebrafish, varies with different angles shown in the right and left eye. As you can see, the central portion of this surface, corresponding to 0, 0, is where the response is maximum. And this dies down as the stimulus becomes more backward, or the points marked by 180 and 180. And this is what you would expect. On the right, I'm showing you the variation of the right weight of the Gaussian. What can we observe over here? The peak of this surface is occurring at more rightward stimuli, which correspond to a 90-degree direction shown in each eye. And the right weight dies down when there's more leftward stimulus. And these trends are what you would expect in real life. As the stimulus is more rightward, the right weight will increase, and as the stimulus tends toward leftward, this weight component will decrease. Well, can we then build a mathematical representation of these surfaces so that we can then characterize the entire optomotor response across the binocular space? And so initially we thought out how to build a model for integration of information from both eyes. So we had the behavioral aspect where we show directed motion cues to both eyes. At the same time, we will be able to conduct experiments where we show directed motion cues to only a single eye and see how these combine. At the behavioral model level, we can see how this binocular motion model that we just described can be broken down into the individual components where we only look at the model for the stimulus presented in the left eye or the stimulus presented in the right eye. And then again, at the neural circuit level, we can see whether the responses generated by the left portion of the brain can be combined with the responses generated from the right portion and combined to form the overall responses. So essentially, in all these levels, we're trying to address the question, how do they combine? And this ties in pretty well with the 3 levels of David Marr that Florian talked about earlier. And this is how we go ahead to address information integration from both eyes in the context of the optomotor response.
The next computational model addresses rheotaxis. As Florian beautifully explained, when the animal is in a stream, the fish tends to orient itself against the flow direction to stabilize its position. This function doesn't require visual input. In the dark, it uses tactile sensors, the neural masks, in the natural line system. Our research questions are 2. First, what is the neural basis for rheotaxis behavior? At the moment, we know relatively little about underlying neural circuits. What we are particularly interested in is to find out specific network motifs that will characterize neural circuits underlying rheotaxis, and to use these motifs to look in the connectomics of the fish to find out the relevant neural circuits that exhibit those motifs. With regards to the fish behavior, as you will see in a moment, this function is relatively challenging, and the fish doesn't always accomplish the goal. So we ask, is the fish rheotaxis response optimal? And if not, if there are algorithms, mathematical or computer algorithms, that can do better than a fish, we would like to know what are the constraints that lead to the observed suboptimal fish behavior. We're also interested to know whether the fish shows adaptive behavior in rheotaxis. And can we design new experiments to find out the extent of adaptive features of rheotaxis? This research will be described in a moment by Nimrod Shachar. Nimrod is a talented young aspiring theoretical neuroscientist who, as a postdoctoral research fellow, develops models of both the behavioral and the neural algorithms underlying rheotaxis function in the fish.
Okay, so as Chaim described, it is a challenging behavior to achieve the goal of rheotaxis, which is not to get carried away by a stream, even without visual information. So here I will describe a behavioral algorithm or a policy that the fish might be using in order to perform this behavior. And then I'll try to describe a neural circuit that can be underlying this behavior. So first, importantly, using the neuromasks, the fish might be able to detect a rotational flow around itself, which is characterized by a curl, a mathematical object that measures how much rotation there is in the flow. And when a fish is found in a stream that is non-uniform, meaning that there is some slope or a gradient in the velocity field, then the fish will feel this kind of a rotational flow, so that the rotation or the curl of the flow is a measure of the gradient or the change, the spatial change of the velocity field. So the behavioral algorithm goes as follows. The fish measure the gradient in two consecutive locations, one before a bout and one after the bout, and it checks the magnitude of the gradient. If the gradient increases, the fish will bias the next bout to be in the direction of rotation, meaning clockwise or counterclockwise. While if the gradient decreases, the fish will generate an unbiased bout centered around its current direction. This kind of behavioral algorithm gives rise to the behavior that we actually see in the fish, which is swimming towards the center of the pipe. And the fish orients itself against the flow. And so now we can ask ourselves, what kind of neural network can generate this kind of algorithmic behavior? So here's our suggestion. We are suggesting the following circuit. As you see in the left picture, you have those R units, which get inputs from the neuromasks in both sides of the fish. R1 unit will be active when the fish feels a counterclockwise rotational flow, and R2 will be active when the fish feels a clockwise rotational flow. So the R's serve as curl detectors, and they can measure the current curl and so the gradient of the flow. And then this information is sent to the M units, which are going to make the decision of whether to bias or not to bias the following bout. But this information goes through a temporally derivative circuit, which is composed of the G units. Those units have a different time scale. One is an inhibitory unit that has a slow time scale, and the other one is excitatory unit with faster time scale. And those units integrate the information from the curl detector, from R, and they will pass the information to the M unit, which will be affected by whether the gradient increased or decreased. If gradient was increased, the M unit will tend to make a decision to bias the bout in the direction of the rotational field. And if the gradient decreased, the next bout won't be biased. As you can see here, this kind of a model, when we simulate it, we simulate fish behavior using this network, generates the very similar statistics of bouts to what we see in the real fish. Here to the left, we see data from real fish. The blue curves are the statistics of bouts when you don't have the gradient decreased, which are quite symmetric. And in red, you see the statistics of bouts where after an increase in gradients, and you see that there is a bump in some positive direction, in the direction of the rotation. This is the bias that the network generates. In addition, we can look at fish trajectories, simulated fish trajectories, and you can see in the top panel that the X location of the fish, which is the perpendicular direction to the flow, tends to go. The fish tends to go to the center of the flow, to X equals 0, and the direction of the fish relative to the flow tends to go to a pi or a pi radians or 180 degrees, which is against the flow, or the fish orients itself against the flow. So here, this is to show that the simulated fish does perform the behavior expected, shown by real fish. And now it will be interesting to check whether we can find these kind of motifs, this kind of inhibition circuit that includes excitation and inhibition, and done by different units in the brain. And these kind of motifs we'll be looking for in electron microscopy. Next, I want to address the optimality of the Rheotaxis behavior. From the algorithm I described, you get two interesting consequences. One, in the regular flow in a stream, as in the top panel here, the fish, as we said, tend to go to the center of the pipe and orient itself against the stream. This is the correct direction to be in, where if you don't want to get carried away, but it's the wrong location, because this is where the stream velocity is maximum. On the other hand, if we can invert the velocity profile so that the maximal velocity is on the side, then the algorithm will take the fish towards the center of the stream, which is the right location, because there the velocity is minimal, but it will be in the wrong direction, because now the fish will face the direction of the flow. So we can ask ourselves whether the fish can do better, given the inputs it gets, the gradient of the flow measured in 2 consecutive places. Can the fish do better than that in not get carried away by stream? In order to address this question, we model the fish motion as a dynamical system. Here you see we use X as the location of the fish perpendicular to the stream, Z as the fish location in the direction of the stream, and theta, the angle, the orientation of the fish compared to the stream. And here we model this policy, the decision-making of the fish, by this function f that gets 2 inputs, the current gradient and the gradient in the 1 time step before. And it will generate a bias of the turn, whether the turn will be, what will be the average direction of the next bout. And now we would like to see what function f will be optimal to achieve the least amount of getting carried away by the stream. In order to optimize this, we use the deep neural network to model this function, the bias function. This is a modern approach used quite a lot now in machine learning and AI research. And we are optimizing this function f to get the minimal amount of drift by the stream. And the results, as you can see here, are that the optimal deep neural network function does pretty similarly to the fish algorithm that we described earlier, meaning that the fish gets carried, the algorithmic fish gets carried away quite similarly to what the network produces, and also the behavior is quite similar. And interestingly, how well the fish can do is restricted to a range of velocities of the stream, which is a point that we would like to be able to test in future experiments. What if we push the fish into regimes of higher velocities than what he can handle, or if we use different interesting profiles and see how similar or different the fish does compared to our model deep neural network. Okay. So to summarize, we showed that we propose a neural circuit model that we can underlie Rheotaxis behavior, and we would like to see if the motifs that we see there can be measured in electron microscopy. And in addition, we use deep neural network to find an optimal policy for the fish to achieve minimal drift in the stream. And we went to validate it by behavioral experiments, and to get out of those models some limitations on what the fish can or cannot do, and then test them again in new behavioral experiments. Thank you. The next speaker will be Jeff Lichtman, who is the head of the electron microscopy research done on the zebrafish, and he will describe the research done in that direction.
My name is Jeff Lichtman, and I'm going to describe for you the automated, I should say mostly automated, connectomics approach we take with serial electron microscopy. We've developed a pipeline to generate a wiring diagram of the larval zebrafish. And this gets to that third level of Marr, where we're getting to the actual implementation in the neural circuits. I want to talk a little bit about this pipeline because although it's a technical problem of generating this wiring diagram, it's taken a great deal of time to do. And so I thought it would be useful maybe to emphasize some aspects of this. The first is that there are many technologically challenging steps to get something to work on the scale of a whole fish. And these steps are not the same. It's not that you have to solve one problem, you have to solve many different problems. And every one of these steps are still under continuous scrutiny to improve their reliability and their speed. All of them could be more reliable, and all of them could be faster. And speed and reliability are the essential ingredients of a successful pipeline. Third, it's nice to know that over the course of this U19, we have, by necessity, actually changed enough things to have seen dramatic improvements in both the throughput and the consistency of our results. We're very grateful for that. It gives us confidence that what seems heroic will soon seem routine. It's not yet routine, but we hope eventually this will be truly routine to do this kind of work. And I hope you'll agree that we've made good progress in generating a full resolution fish connectome. So let me talk a little bit about the pipeline. Pipeline is a kind of funny term, but it means that we're passing the fish from one procedure to another, and ultimately we get rid of the fish, and then we deal with the digital data, and that is what carries us all the way to the finish line, which would be a complete analysis, I guess, of the wiring diagram of an animal. I'm going to go through the many steps of this pipeline somewhat briefly, but just to give you a sense of how many things have to work in order to get a fish wiring diagram. To start with, we don't just take a fish right out of an aquarium or out of a river. We take a fish that is often already a transgenic animal that expresses either calcium indicators or perhaps labels, molecular labels, that give us some insight into the fish and that are visualized either by two-photon microscopy or confocal microscopy. The fish that we've been working on and that we will describe in detail for you in the next 15 minutes or so is a fish where the information was not activity information, but functional information of a different sort. We've divided the fish into cells that are excitatory and cells that are inhibitory, and those were imaged with a confocal microscope. A living fish was imaged that way, and those two sets of cells were labeled different colors. And that's the fish. You'll hear much more about that from Mariella, who will speak after me. That's the fish that we went on and have been working on its connectome. So first we have a fish, then we have to preserve the fish, and that first step in preservation is to fix it, to put aldehydes in there to basically harden it and stabilize the structure. That's not as trivial as just dunking the fish in fixative, because the fixative won't go through the skin. So a great deal of effort has gone into making good fixation by making a narrow slit in the fish to expose parts of the animal to fixative that would otherwise be protected from it by the skin. And then following the fixation step, we then have to metallize the fish, turn every single membrane into something that's visible in an electron microscope. And that requires staining the fish with heavy metals, in particular osmium, which is a very good electron microscopy signal. But again, it is not trivial to get a fish osmicated to the point where all the cells are well labeled. After the labeling, then we have to infiltrate the fish with a heavy, hard resin, a thick resin that's very hard, which would allow us now to slice the fish in very thin sections without having it to crumble. So resin infiltration is the next step, and we trim that block. And then in some fish, now it's de rigueur to do this, but in this first animal, we haven't done this, but I'm just going to mention this because it's now part of the pipeline. We image the entire fish in a micro CT machine, a computerized tomography machine using X-rays. That gives us cellular level resolution of all the cells of the nervous system, in fact, all the cells in the body. In addition, it tells us whether the stain was successful, and it provides a very nice intermediate point between the optical imaging that was done with a confocal or 2-photon and the ultimate electron microscopy imaging, which is done after the sectioning. So it's a nice point for everything to be aligned to the micro CT image. Once we feel the block is useful and it's worth going on, we then cut the block into many, many thin sections. We use a automatic tape collecting ultramicrotome, that's what ATOM stands for, which basically means we're cutting the fish very thin, 30 nanometer sections, and picking these sections up on a tape that collects all of the animal one section at a time. So you take a three-dimensional fish and linearize it into a bunch of little sections, and then those sections are put on tape, and that tape ends up on reels. And then the tape is cut into strips, and those strips are pasted onto silicon wafers that are very flat. And you can see in that middle picture a series of strips with little gray spots on them. Each of those spots is a separate section, and those wafers are what ultimately will be imaged in the electron microscope. But before imaging a wafer in the electron microscope, we have to decide where the sections are on those wafers, because it would waste a lot of electron microscopy time hunting around for each section. So we then do another kind of imaging. We take the wafer and a stage holder, and we image it with a reflected light optical microscope to localize every single section on each wafer. And then we have what we call it is a mapped wafer, where each section is position is known. And then that plus the stage holder is put into a remarkable new machine, a multi-beam scanning electron microscope that was designed actually for connectomics for this project. The first one ever is in our laboratory. That is what you see a picture of it there. It's a very fast machine that's designed for rapid imaging of large volumes by fast imaging of section after section with a multi-beam. I'll say a little bit more about that. And in order to make the multi-beam super efficient, which it wasn't when we first got it, we built a workflow manager that allows us to, in real time, make sure it's taking images of high quality and also providing us with thumbnails, smaller images of the whole data set that are more tractable than the 350 terabytes this data set is altogether, that we can use for alignment, and you'll see a lot of those images coming up. And then we can register those thumbnails, which have cellular resolution, with the optical microscopy. You'll hear Mariella talk about that, so I won't go into much detail about that. And at the same time, we do a full resolution stitching and alignment, which is computationally intense. And then with that, we can then generate, with that full resolution data, ground truth. We can trace out every single wire in a small volume and do that in several different volumes. And that provides information for machine learning algorithms that allow a team, in this case, Google, to segment the whole fish, every single wire being figured out just based on a small amount of ground truth. And so ultimately, we have two kinds of data. We have the functional data and the segmentation data. And of course, we have all the theory and expectations based on theory to allow us to do some kind of circuit description and relate it to an analysis to validate or test or constrain theories that have been coming up. So I just want to briefly mention a few things about the fish in particular. The first is that the sectioning of a whole fish is a lot of sections. 30 nanometers is 1,000th as thick as a human hair. So you can imagine you have to cut a lot of sections. And indeed, this 7-day post-fertilization larval fish was first trimmed and cut in a longitudinal direction. That is, the sections were cut so from head to tail, each section, and nicely symmetrical left and right side. And we collected 17,500 sections at 30 nanometers. Richard Schleck did most of the cutting, and he's part of the team here, really amazingly good at doing this kind of cutting. And then that is a picture of the fish there. The dotted line on the bottom and the dotted line to the right is what wasn't cut, but everything else was cut. And that generated 3 reels of tape with sections on it. And now those tape have all been turned into wafers. And on the top of this image here, you see that a whole zebrafish brain has been turned into a bunch of petri dishes, 89 wafers with 12,500 sections. And on the bottom is the part of the fish that has now been put onto sections. Now, we haven't imaged all those sections at this point. At this point, we've only imaged 6,579 contiguous sections on 48 wafers, but that is the part that is mostly nervous system. 80% of the CNS is in those 48 wafers, which are each shown there. And that totals about 200 microns in depth, and all of them have been mapped, as I've described, and imaged in the multi-beam electron microscope at 4 by 4 nanometers in the lateral plane, and each section is 30 nanometers thick. So 4 by 4 by 30 nanometers, or 480 cubic nanometers, which is a very small voxel, if you will, and that generated 350 terabytes. So the multi-beam, I just want to say 1 or 2 words about it because it's such an interesting invention, and it has certainly helped a lot of connectomics moving forward. It's a high throughput machine because instead of having 1 scanning beam, it has 61 beams scanning in parallel, and these 61 beams generate a multi-beam field of interest, basically. These MFOVs are 61 beams and arranged in a hexagonal way. And so if you have a bunch of them together, you can generate, by tiling, a very large area. Each image section is somewhere between 50 and 100 gigabytes, depending on how much brain is in that section. And we generate for 1 section somewhere between 100 and 200 of these MFOVs have to be stitched together. Here's shown 7 MFOVs stitched together, and that generates something that's about 300 microns by 300 microns. Each of these MFOVs is a hexagon, as I've mentioned. Those are the 61 beams, and it's about half a gigabyte and each one is made up of 61 stitch tiles that are visible there, and that generates about 100 microns of data simultaneously. So that's the unit of image capture, a field of view of an M file, multiple scanning field of view, and each of the tiles in there is about 8.5 megabytes, 3 by 3,000 by 2,700 pixels. So they're still very big images. Each tile is a very big image, but this whole imaging adventure so far has generated 40 million such tiles. And here's what one of those tiles looks like. And they are good enough to see synaptic vesicles, so they're very high resolution. One other thing, and the last thing I want to say, is that to get through the data, if you have to do something 40 million times, it doesn't mean every single time it works. So you have to have some way to manage the data coming through. And we've gotten high throughput thanks to work Yulong Wu, another member of the team, has done making a real-time MSEM workflow manager, which assesses image quality with numerous checks validating the files. It also includes spreadsheets and color-coded tiles that itemize the quality of every tile and easy clickable access of any data that needs scrutiny. This picture you're seeing, for example, has a pinkish area which has no interesting data in it at all. It's actually just resin, and that's why it's pink, because there's nothing in focus there, because there's nothing to see. The areas that are green are areas that have high density of membranes, and any place that is not green but should be green, we can click on and see whether it's because there's a staining artifact or the machine was not in focus if we have to retake anything. And finally, it generates aligned thumbnails of the acquired data, which are really valuable, as you'll see going forward. So I'm going to hand the reins over to Mariela Petkova, who actually has masterminded this entire workflow of the fish that we have gotten through the pipeline, thanks to her. And it'll give you a sense of how far we've gotten to get to the wiring diagram.
Hi, I'm Mariela, and I'm here to help you visualize how we're going to go from connectomics to actually testing circuits. What you see on your screen right now are two rotating brains, and in those brains I have labeled the sensory input and the motor outputs for the fish. What is really exciting about doing connectomics in the whole animal is that we can actually see where the signals are coming from and where they're going. And our job is to basically connect the dots. On the left, you see the sensory input for the OMR, optomotor response. If you see in cyan, the two optic nerves, which come from the eyes, and they will arborize in the brain, and they'll start making connections, which we can see if the connections they make match what the model predicts. In green, you see two very big neurons. Those are called the magnet neurons. They are the motor command neurons, and basically they send motor signals and activate the movements of the fish. Of course, they don't act alone. There are quite a few other neurons, but this is mostly the motor center for the fish. On the right, you see the Rheotaxis sensory inputs. Those are the so-called afferent lateral line neurons. They send signals from the tail, from sensory neuromass along the tail, all the way to the brain. And in the next slide, I will focus on the very first synapse that these neurons make in the brain. So what you see here on the right are the two lateral line afferent neurons. They're in shades of blue. Those are the axons, so they send information from the tail to the brain. And we can see the very first synapses that they make with dendrites in the central nervous system. Those central nervous system neurons are shown in purple and in pink. And just based on the connectivity, what we can see is, for instance, the pink neuron really hugs this dark blue neuron, afferent axon, and it makes multiple recurrent connections with this axon. What this tells us is that there is a very strong connection between those two neurons. If you take a look at the purple neuron, on the other hand, that neuron connects with both the light blue and the dark blue lateral line afferent. It means that it's receiving signals from both neurons, and that is basically a way to integrate the signals that come from those neurons. What you see here are 3D structures, and those 3D structures come from electron microscopy data. In the next slide, I'll show you a movie of the whole fish. I have labeled for you the larger anatomical structures, and in one inset, you will see a high-resolution image of all of the axons and dendrites that are inside a very small portion of the high-resolution image. So let's take a look. What you just saw in the upper right corner was an inset which probably looked to you like a hopeless tangle of membranes and neurons, and you couldn't tell it apart. Here I'm showing you something similar, which is a little bit easy for you to distinguish. On the left side are the raw uncolored images from the EM data. In very dark, you see the membrane stained, and in the very big dark circles that you see are myelin actually stained neurons. On the left side, we have colored in each object individually, and you can already imagine what this structure looks like in 3D. I'm guessing that you already can see that you're literally sliding down a bundle of neurons. And I can show you the structure in its 3D shape. To color in this little box, which is 4 by 4 by 8 microns, it took us about 200 hours. Now, the whole thing is way, way bigger, and it will take more than a lifetime for a human to color in every single object. So as Jeff mentioned, we're training machine learning algorithms in order to color the pictures for us. And the way that we have done this is we traced and colored in 6 boxes from different places in the fish. As you can see, just by looking at the objects, they're very different regions, and they have very different ways of looking. And we use those 6 boxes to teach the computer how to color in for us. Next, we show the computers just blank canvases that they haven't seen before. And what they did is they colored in all of the objects for us. And this is really exciting because if you look through the whole coloring book of the brain, after it has been colored, you can just pull out the structures that exist. And this is an example of one of those structures, actually two. What you see on the right side are the 3D shapes of a dendrite and an axon. The dendrite is in blue, the axon is in green. You can tell, maybe if you know and suspect that dendrites have spines, that this is a dendrite, but we can actually really confidently say which part is an axon and which part is a dendrite from the EM images. Take a look on the left. What you see there in green is the axon that I'm talking about. And the reason why I know it's an axon is if you look at the little tiny circles that are inside the green shape. Those are vesicles which are full with neurotransmitter. So you know that when a nerve impulse arrives, these vesicles will be released, and they will travel through to the blue-colored dendrite. You see that there is a very tiny gray region in between. That's where the junction between the two neurons are. So we can use the EM images to give us the structure of the neurons, to give us which neurons talk to each other, and also the direction of this of the connection between the neurons. What we don't know yet is whether or not we can tell if a neuron is excitatory or inhibitory. And we really need this type of information in order to verify the models, because any circuit model really uses information about excitation and inhibition. And in order to do that, we're using the fact that the zebrafish is a transparent animal, and it's very easy to do transgenics with it. So we have labeled the excitatory and inhibitory neurons, and we have imaged them with a confocal microscope. So what you're seeing here are 3 rotating images of the same animal. On the far right, you should recognize the EM image. And on the left side, you see the confocal images of the excitatory neurons in magenta and the inhibitory neurons in cyan. There are, of course, many other neurons in the brain that we have not visualized, and but those will show up as gray when we overlay the 2 images. So what I'm going to do next is I'm going to show you the confocal stack of the animal, and I'm going to overlay it on top of the electron microscopy. What you will see is that all of the excitatory neurons will be overlaid in magenta, all of the inhibitory neurons will be overlaid in cyan, and the rest of the neurons, which are gray, we do not know what kind of genotype they have. And what is really exciting about being able to do this in the same animal is that we can very confidently say that this cell, we can very confidently find a cell in the confocal microscope and the EM microscope, and this will tell us what kind of cell it is. So let's take a look at the images, the movies. What you just saw was the overlay between the confocal images and the EM images. Now what I'm showing you is I'm zooming in so you can really see the cells and the overlap between the 2 data sets. I have inverted the colors of the EM so that you can see the membranes of each cell as these white circles, and the excitatory and inhibitory neurons are shown in magenta and in blue. What you should see is that each circle is already colored, and you don't see mixing between the purple and the blue colors, which means that we can really confidently say which neuron is excitatory and which one is inhibitory. And with that, I'll finish. Thank you so much. Thank you for listening, and I'll hand it back to Florian, who will give some concluding remarks.
So you've heard now, lastly from Mariella, how we can use the combination of light microscopy and electron microscopy to get a first shot at identifying neural circuits in the larval zebrafish's brain that are actually doing something important. And I want to use this opportunity to take up again the question of the curl detector, because that's, I think, where we have a complete picture now, a complete problem. The fact that fish swim against the current argues, along with Marr level 1, that it's important for them to stay stable in a moving stream. The algorithm, especially the Rheotaxis algorithm, as you've probably appreciated, is not trivial at all. It's a complex computation that this animal needs to do, that involves complicated sensory processing and important computations. So it's a challenging problem. I think we have made great headway there. And as you've appreciated from Mariella's presentation, we even now have a concrete hypothesis on what elements, what neural circuit elements could be involved, and why connectomics and EM-based circuitry analysis is really a very useful and powerful way of constraining and validating these circuit models. So I think we've sort of come full circle here with this particular example, and I hope it illustrates what a powerful technique all of these approaches manifest, and how we can use the combination of theory, behavioral imaging, calcium imaging, and connectomics to really make headway in the question of how small vertebrate animals solves an important adaptive problem. And with that, again, I'd like to thank all of the team members who have contributed so significantly to the whole project. And I also would like to thank the audience for their attention.
Thank you for that outstanding presentation. For additional panels on the NIH Brain Initiative, view our agenda on our events page. And as a final reminder, the panelists will follow up with any questions you've submitted via email. Thank you again for your participation.